Hello friends, today we are going to study about motifs, domains and folds. Now what are motifs? Before understanding about the motifs, what is the difference between a 2D structure and a 3D structure? 3D structure is the assembly of 2D structure. For example, as we already studied, like alpha helix is a 2D structure. The combination of the assembly of two alpha helix is a 3D structure. So 3D structure is an assemblage of 2D structures, and these secondary structures are connected with segments. Now, there is a difference between fibrous protein and globular protein. Apart from the differences we studied, if we look into the structure of a globular protein, if this is a globular protein, then clearly we can see that it has some interior portion which is separated from the external aquatic environment and an exterior surface which is in connection with aquatic environment. So in case of globular protein, it has an interior segment which is well separated from the aquatic environment, and it has an exterior surface which is well connected with the aquatic environment. Because of this, there occurs a proper distribution of all the amino acids in a globular protein. So what these distributions are? First, the nonpolar residues. The nonpolar residues like valine, leucine, etc. These are mostly occur in the interior of the protein, which is not in connection with the aquatic solvent or aqueous solvent. Second is the charged polar residues like arginine, histidine, etc. These are located on the surface of the protein. And the uncharged polar groups, they are found usually on the protein surface, but it can also occur in the interior of the molecule, because though they are polar, but they are uncharged. So they can found on exterior side also, as well as on the interior side of the globular protein. Now the turns and loops. When we are saying that 3D structure is an assembly of 2D structure, so if 2D structure is present, definitely they are connected with some loops or turns. So these loops and turns are usually occur at the protein surface. Now what are supersecondary structures? Supersecondary structures means it is a stable arrangement of many secondary structures. These are also known as motifs or folds. This can be simple or it can be a complex. Sometimes an entire protein can be a supersecondary structure. For example, coiled coil of alpha keratin. So supersecondary structure means it is a combination of many secondary structure. What happens in coiled coil? One alpha helix is arranged in a super twisted manner with another alpha helix. So this is the combination of two secondary structures. So this is a supersecondary structure, which is also known as motifs and folds. Now let's see some simple motifs. One such motif is beta hairpin. What is beta hairpin? It looks like this. Here, this is a beta sheet. This is a beta sheet. But these are parallel beta sheets. So the parallel beta sheets connected with alpha helix. This is a one kind of motif. And then anti-parallel beta sheets connected by turns. This is also a kind of motif. So beta hairpin motif, in this mainly we consider this anti-parallel one only, but this is also a type of motif which we can call at beta alpha beta motif, because two beta strands, parallel beta strands, are connected by alpha helix. So this motif we can also call it as beta alpha beta motif. One more motif is alpha alpha motif. As the name suggests, that means two alpha helix are connected. But how they are connected? With a turn. They are inclined at an angle and connected with each other via a turn. Next is Greek key motif. Greek key motif is nothing but these are also the connection of beta strands, but in a particular way of connection. This is known as Greek key. So in this pattern, when these beta sheets are connected, then they are known as Greek key motif. So it is also a beta hairpin motif only, because here also the beta sheets are connected, and that to the antiparallel beta sheets only. Next is some complex motifs. Complex motifs are the combination of simple motifs. So one such example is alpha beta barrel. So here in the diagram only you can see this is one set of beta alpha beta loop, and this is alpha beta barrel. The connection of these two and the whole is known as alpha beta barrel, which is a very complex motif because it is made up of simple motifs. Now, such type of motifs are seen in many enzymes as a binding site for a cofactor or a substrate. They form a pocket-like structure that helps to bind with the cofactor. Okay, the next is beta barrel. Now, beta barrel is again the combinations of simple beta motifs. We already seen in the last slide that beta motif or beta hairpin loop, in that two antiparallel beta strands are connected by turns. So here multiple such beta hairpin loops are connected in a sequence having different kind of connections. The arrangement of these connections are different in all the three figures. So that such type of motifs comes under a complex motif and the name is beta barrel. So here this type of arrangement is seen in retinol binding protein. The B such type of complex has been seen in peptide N asparagine amidase, and the third one C has been seen in triose phosphate isomerase. So the motifs can be simple one and also can be the complex one, that is the combination of many simple motifs. Now, protein motifs, these are the basis for protein structural classification. Now, protein structures we can divide into four classes. Four classes of proteins are there with respect to their motif structure. The protein can be a type having all the alpha helix, example E. coli cytochrome B562, all with beta motifs, example immunoglobulin folds. It can be of alpha beta or alpha and beta motifs, example is lactate dehydrogenase. So based on the type of motif, the proteins can be divided into four classes having all alpha, having all beta motifs, or having alpha and beta combination of motifs. Example is lactate hydrogenase for the combination, for beta it is immunoglobulin, for alpha it is cytochrome B562. Now what are domains? Domain, a single protein can be made up of multiple domains and each domain can function independently and their function can be different. Therefore, domain is also a kind of motif, but domain means it is the functional subunit or the functional polypeptide of a protein. So what are domains? These are two or more stable globular units of polypeptide. A protein can contain multiple domains. Sometimes these domains are so closely associated that it is become very difficult to distinguish that the protein is made up of single domain or it is having multiple domains. And all the domains often have different functions. There are proteins also that have only one domain. Now example is glyceraldehyde 3-phosphate dehydrogenase. Now glyceraldehyde 3-phosphate dehydrogenase, can you tell me where this enzyme is used? This is an important enzyme of glycolysis. What it do? Glyceraldehyde 3-phosphate dehydrogenase, it oxidize and phosphorylate glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate. So this enzyme, glyceraldehyde 3-phosphodehydrogenase, it consists of two different domains. One domain binds with NAD plus and the another domain is its active site where this conversion takes place. So this enzyme consists of two domains, one for binding NAD plus and another for converting glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate. Here, in case of glyceraldehyde 3-phosphodehydrogenase, two of beta alpha beta unit combine to form a domain, and this is known as dinucleotide binding fold or Roseman fold. So what it binds? It binds the NAD plus. Now one point to remember that many domains are structurally independent unit that have a characteristics of small globular protein. That means individual domain can function as a individual protein. So now one question from my end for you all, that is globular proteins are typically constructed from several layers of secondary structure with a hydrophobic core and a hydrophilic surface. Is this true for a fibrous protein such as keratin? Do let me know in the comment section if you found the video informative. Please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.